Today's topic is stomach emptying, gastric emptying and pancreatic secretion. So about 4 or 5 slides are left of last class about the gastric emptying. So we'll be first completing that. Then we'll be doing gastric emptying. And so gastric emptying, it is the process of the um, emptying the gastric contents from the stomach into the duodenum. Okay? It is the process of moving the gastric contents from the stomach into the duodenum. So the content of the stomach. So यो कति आइरह्यो यार. The content of the stomach. So content of the stomach is the liquid diet, they lives in 10 to 20 minutes, and the solid diet, they remain into the stomach for 1 to 4 hours. Okay. So while the gastric contents or the chyme is leaving from the stomach, it leaves by drop. Okay, liquid. Mainly, it is converted in the form of liquid or semi-solid. Then it leaves from the stomach. Okay, so the particle size, it must be 0.3 mm. Okay? The particle size of the contents that leave the stomach, it must be 0.3 mm. So the composition of the chyme reaching the duodenum, it have high concentration of acid, it consists of fats, it consists of protein, and it consists of carbohydrates. Okay, so neural stimulus such as parasympathetic nerve stimulation, it increases the gastric motility. Right? Parasympathetic stimulation increases the gastric motility or gastric emptying, whereas the sympathetic nervous system, it decreases the gastric emptying. Right? Parasympathetic nerve, what it does, it increases the gastric emptying, whereas sympathetic nerve, it decreases the gastric emptying, because parasympathetic nerve, it causes the relaxation of the sphincter. Okay, parasympathetic nerve causes the relaxation of the sphincter, whereas sympathetic nerve causes the contraction of the sphincter. Okay, pyloric sphincter, it causes the contraction of pyloric sphincter, sympathetic nervous system. That is why there is decrease in the gastric emptying, whereas parasympathetic nerve, it causes relaxation of the pyloric sphincter and it increases the gastric emptying. And there are different hormones which act on the gastric emptying. Okay, so secretin, cholecystokinin, peptide YY, gastrin, and somatostatin, they all inhibit the gastric emptying. Okay, they decreases the gastric emptying. Right? All of these hormones decreases the gastric emptying. So this gastric emptying, it is determined by, we have already told about the gastric emptying. It is determined by the composition of the food. The liquid foods, they empty first, then the solid foods. Fine foods, they empty first, then the coarse foods. Osmolality foods, they empty first, then the non-osmolality food. Then sugars, they empty first, then proteins, then fat. Okay, liquid first, liquid foods, finally churned foods, osmolality solutions, sugars, they mainly empty first. Okay, they mainly empty from the stomach at first. This is the figure of the gastric emptying. Okay, so this is the gastric emptying. It is affected mainly by the two nervous system, neuronal and hormonal. Okay, gastric emptying is regulated by neuronal as well as hormonal. So sympathetic nervous system, it decreases the gastric emptying. There is the positive sign here. That means it will stimulate. It decreases the gastric emptying. Whereas parasympathetic nervous system, it increases the gastric emptying. secretin, it decreases the gastric emptying. Okay, secretin is produced in response to the acid in the duodenum, acidic chyme in the duodenum. Okay, so secretin is produced in response to acidic chyme in the duodenum, and secretin decreases the gastric emptying. Right? Then cholecystokinin and gastric inhibitory peptide, they are released in response to fat and proteins in the duodenum. Okay, fats and proteins in the duodenum, and it also decreases the gastric emptying. If there is hypotonicity, that also decreases the gastric emptying. And gastrin itself inhibits the gastric emptying. Okay, it decreases the gastric emptying. Right? So gastric emptying is mainly affected by neuronal and hormonal. Okay, just remember this: neuronal, hormonal. Neuronal is through the autonomic nervous system, sympathetic nervous system, and parasympathetic nervous system. Okay, neuronal is through the two. One is the sympathetic nervous system and parasympathetic nervous system. Sympathetic nervous system, it decreases the gastric emptying. Okay, it decreases the gastric emptying. Whereas parasympathetic nervous system, it increases the gastric emptying. And there is secretin. Secretin, it decreases the gastric emptying. There are different hormones, mainly four hormones. Okay, mainly four hormones: secretin, cholecystokinin, gastric inhibitory peptide, and gastrin. Right? These four hormones: secretin, cholecystokinin, gastric inhibitory peptide, and gastrin. These four hormones inhibit gastric emptying. Okay, so this secretin, cholecystokinin, gastrin, gastric inhibitory peptide, they all are transported through the blood, and they will act on the stomach. Okay, they all are transferred through the blood. They are not going through the lumen. Right? Just remember this: they are not, these hormones, they are not going through the lumen. They are secreted by the cells present in the duodenum. Okay, they are secreted by the cells present in the duodenum, and they are absorbed into the blood. From the blood, they are taken into these cells. Okay, they are taken there, and gastric emptying is decreased. Right? And gastric emptying is decreased. So this is the process of gastric emptying. Right? This is the process of gastric emptying. So gastric contents, they move in response to neuronal and hormonal. Okay, gastric contents, they move in response to neuronal and hormonal stimulus. Sympathetic nervous system decreases, parasympathetic nervous system increases, other hormones increases. Some of the hormones also increases the gastric motility, such as motilin. Okay, motilin increases the gastric motility. So when gastric motility is increased, that means there is the pressure in the duodenum, stomach is increased, and in the then the pressure in the duodenum. Okay, pressure in the stomach is increased, then pressure in the duodenum. So gastric contents, they move drop by drop. Okay, they move drop by drop. They empty their contents, and while emptying, same neuronal and hormonal mechanism works. Okay, same neuronal and hormonal mechanism works. So sympathetic nervous system decreases gastric motility, parasympathetic nervous system increases gastric motility, and other hormones that decreases gastric motility are secretin, cholecystokinin. Okay, cholecystokinin, gastric inhibitory peptide, and gastrin. These all inhibit the gastric emptying or stomach emptying. This is about the gastric motility and emptying. This is about the gastric motility and emptying. Now we'll be reading the, we will be doing pancreatic secretion. Okay, pancreatic secretion will be doing. Am I clear about the gastric emptying?
Yes, sir.
Right, there are two mechanisms of gastric emptying. One is the neuronal, another is the hormonal. Okay, neuronal is sympathetic and parasympathetic. Okay, neuronal is controlled by autonomic nervous system, that is sympathetic and parasympathetic nervous system. Sympathetic nervous system decreases the gastric emptying, whereas parasympathetic nervous system increases the gastric emptying. Right, there are different hormones. Mainly four important hormones are there. One is secretin. Okay, secretin is released in response to acidic chyme in the duodenum. Right? Secretin is released in response to acidic chyme in the duodenum. When acidic chyme reaches the duodenum, then secretin is released. Secretin inhibits the gastric emptying. Second is the cholecystokinin. Cholecystokinin, they are cholecystokinin. It is released in response to uh, it is released in response to the uh, fats in the duodenum, fats and proteins in the duodenum. Okay, cholecystokinin is released due to the fats and presence of fats and protein particles or and amino acids in the duodenum, peptides in the duodenum. So it goes through the blood, cholecystokinin, and it acts to decrease the gastric emptying. And there is another hormone, gastric inhibitory peptide. It also decreases the gastric emptying. And gastrin, it also decreases the gastric emptying. So liquid particles, they empty first. Finely churned particles, they empty first. And the protein and the sugar, they empty first. Okay, osmolality solution, they empty first. So this is all about the gastric emptying. So now we will be seeing the pancreatic secretion. We will be doing the pancreatic secretion. So we ingest water, there is the secretion of the saliva, there is the secretion of the gastric contents, and there is the secretion from the pancreatic juice and bile. Okay, pancreatic and bile juice also secrete. And small intestine, it absorbs about 700 ml of the water per day, right? Ml of what we ingest per day. So the final product of the cephalic and the gastric phase is after the cephalic and gastric phase in the stomach. Okay, after cephalic and gastric phase in the stomach, there is the digestion of the proteins, there is the formation of the chyme, and there is the controlled entry of the chyme into the intestine. Right? This is the gastric emptying. Right? First, there is the digestion of the proteins, there is the formation of the chyme, and there is the controlled entry of the chyme into the intestine. Until and unless the solid food particles are not converted into the smaller particles, they will not be sent to the duodenum. Okay. So for that, there is the neuronal loop and there is the hormonal loop. I know there is the neuronal feedback control and there is the hormonal feedback control. Neuronal by sympathetic and parasympathetic nervous system, hormone by gastrin, secretin, cholecystokinin, and gastric inhibitory peptide. Right? So the hormonal and neural aspects of the intestinal phase. So upon entrance of the chyme into the duodenum, it gets the enteric nervous system activated, and that secretes the secretin. So secretin slows the gastric emptying and gastric acid production. It stimulates the bicarbonate production so that um, it stimulates the bicarbonate production from the pancreas so that the acidic chyme which arrives from the duodenum, which arrives from the stomach, they will be neutralized. Okay, if the secretin is not produced, if there is no pancreatic secretion, if secretin is not produced and if there is no pancreatic secretion, then the acidic chyme, it will erode the duodenal mucous membrane and it will cause duodenal ulcer. Okay, so it is important. Right? So secretin is also the first hormone known. Okay, first hormone known is the secretin. Right? Then there is the cholecystokinin. Cholecystokinin, cholecystokinin, it is secreted in response to the lipids, okay, fats, and in response to the peptides. When fats and peptides are comes into the duodenum, and the cholecystokinin hormone is released. Cholecystokinin hormone, it slows the gastric motility and gastric acid secretion. Then there are other hormones also, gastric inhibitory peptide and glucagon like peptide. So gastric inhibitory peptide, it inhibits the gastric emptying, whereas the glucagon like peptide, it increases the insulin release from the pancreas. Okay, gastric inhibitory peptide, it slows the gastric acid and gastric emptying, whereas the glucagon like peptide, it stimulates the insulin release from the pancreas also. So the major process that occur in the intestinal phase are mainly the buffering. Okay, because the acidic chyme which comes into the duodenum, it may erode the intestinal mucous membrane. Okay, it may erode the intestinal duodenal mucous membrane. it may cause the duodenal ulcer. So there is need of the buffering action. Okay, there is the need of the buffering action, and this buffering is done by the release of bicarbonate from pancreas. Okay, buffering is done by the release of bicarbonate from the pancreas, and pancreas secretes bicarbonate in response to secretin. Okay, pancreas secretes bicarbonate in response to secretin. So this buffering action is through the exocrine secretion of the pancreas, right? So after buffering is being done, there is the digestion occurs. Okay, in the small intestine, there is the digestion occurring. So digestion is by the pancreatic exocrine secretion. So these pancreatic exocrine secretion, they release the proteolytic enzyme, they release the lipolytic enzyme, they release the carbohydrate digesting enzyme. Okay, they release the pancreatic lipase, they release the pancreatic alpha amylase that digests the sugar. Pancreatic lipase digests the fats, and trypsinogen, chymotrypsinogen, procarboxyl peptidase, procolipase. These enzymes, they um, digest the proteins. Okay, pancreatic exocrine secretion, it consists of carbohydrate digesting enzymes, fat digesting enzymes, and protein digesting enzymes. Carbohydrate digesting enzyme is the pancreatic alpha amylase. Fat digesting enzyme is pancreatic lipase. Protein digesting enzyme is trypsinogen, chymotrypsinogen, procarboxypeptidase. Okay, these are the carbohydrate digesting enzymes, and these are the protein digesting enzymes. Okay, trypsinogen, chymotrypsinogen, procarboxypeptidase. These are the protein digesting enzymes. So these enzymes, they are in the inactive form. Okay, these trypsinogen, chymotrypsinogen, procarboxypeptidase, they are in the inactive form. They will be converted into the active form. They are converted into the active form by enteropeptides that is present in the gross border of the small intestine. And they are converted into the active form. And the digestion, it also occurs. Fat digestion also occurs by the release of bile from the gallbladder. Okay, it also occurs by the release of bile from the gallbladder. So cholecystokinin is again responsible for secretion of the bile from the gallbladder, right? So cholecystokinin, it, the pancreatic exocrine secretion, okay, these enzyme secretion is done by cholecystokinin also, right? Cholecystokinin, it increases the pancreatic secretion which is rich in enzyme, right? Cholecystokinin causes pancreatic secretion which is rich in enzyme. Secretin causes the pancreatic secretion which is rich in bicarbonate. Remember these two things, right? Cholecystokinin causes bicarbonate. Cholecystokinin causes enzymatic secretion which is rich in enzymes. Okay, cholecystokinin causes secretion which is rich in enzyme, pancreatic secretion which is rich in enzyme, whereas secretin causes pancreatic secretion which is rich in bicarbonate. Okay, it causes secretion which is rich in bicarbonate. And cholecystokinin also, uh, cholecystokinin also causes the release of bile. Okay, it causes the contraction of gallbladder and relaxation of sphincter of OD. Right, cholecystokinin causes the contraction of gallbladder and relaxation of sphincter of OD, so that the bile is released into the second part of the duodenum. Okay, bile is released into the second part of the duodenum. So bile, it helps in emulsification of lipids. It increases the area for the pancreatic lipase. Okay, it increases the surface area for the pancreatic lipase, bile salts. So there are cross border enzymes which peptidase, enteropeptidase, which also helps in digestion of the proteins. Okay, and finally there is the absorption in the small intestine, right? Buffering action, digestion, and absorption. So most of the water and nutrients, they are absorbed in the small intestine. All the water and nutrients, they are absorbed in the small intestine, right? Most of the water and nutrients, they are absorbed in the small intestine. So look at this figure. Okay, so there is the food into the stomach. After the arrival of the food in the stomach, there is the acid secretion, there is the pepsin secretion, there is the gastric lipase secretion. Okay, so uh, all this secretion, it helps in converting the bolus of the food into the semi-solid particle, that is the chyme. Okay, right? Food is converted into the chyme in the stomach, and once the chyme is totally liquid, then there is the gastric emptying. Okay, there is the motility and there is the gastric emptying. So this gastric emptying, okay, this gastric emptying, it goes into the small intestine. In the small intestine, there the hyperosmotic solution, they go first, carbohydrates they go first, and fats and proteins they leave at the end. Okay, and there is the acidic chyme is also secreted. Once acidic chyme is released into the duodenum, it reaches into the duodenum, there is the secretion of the secretin. Secretin, it increases the pancreatic bicarbonate rich secretion. Okay, and it not secretin, it increases the bicarbonate rich secretion and it neutralizes the acidic chyme. Okay, secretin. Secretin not only neutralizes the acidic chyme, but it also inhibits the acid secretion as well as gastric motility. Okay, these dotted lines, they represent the inhibition. Okay, dotted lines represents the inhibition. So secretin, it causes the bicarbonate rich secretion, pancreatic secretion, as well as it inhibits the gastric motility as well as gastric secretion. Likewise, fats and proteins, in the presence of fats and proteins in the duodenum, it increases the cholecystokinin secretion. Cholecystokinin, it increases the pancreatic enzyme secretion, which is rich in the carbohydrate digesting enzymes, fat digesting enzymes, and protein digesting enzymes. Okay, so there is the digestion done by the pancreatic rich enzymes, and there is the inhibition of the gastric motility and gastric acid secretion, because at a time large volume of the contents or the chyme cannot be digested by the small intestine. There must be proper digestion as well as absorption. So the food particles, they go by drop, right? So it must empty, right? It must inhibit the acid secretion and gastric motility. And there are carbohydrates. Okay, carbohydrates, they cause the secretion of gastric inhibitory peptide and glucagon like peptide, which causes the release of insulin secretion, and this insulin, it helps in absorption of the glucose. Okay, it helps in absorption of the glucose. Okay, so if there is more gastric inhibitory peptide, it again inhibits the gastric acid secretion. So the secretion in the small intestine is mainly from the pancreas. Right? So pancreas have the endocrine part. This one is the endocrine part, and these are the acinus. Okay, acini. These are the exocrine part. Okay, this is the exocrine part. Right? These are the pancreatic acinus. These are the exocrine part of the gland. Okay, so and this is the endocrine part. Okay, pancreatic islets of Langerhans, these are the endocrine part. So pancreatic endocrine part, it secretes the insulin, glucagon, somatostatin. Okay, it secretes the insulin, glucagon, and somatostatin. Pancreatic endocrine part, this is the endocrine part. So it secretes the insulin, glucagon, and somatostatin. Whereas the exocrine part, it secretes the enzymes. Okay, this is the exocrine part. Exocrine part consists of the acini and ductal cells. Like in the salivary gland, there are the ductal cells and acinar cells. So in the pancreas also, there is the acinar cells and the ductal cells. Right? In the pancreas also, there are the acinar cells and the ductal cells. So these acinar cells, they release the enzymes of the pancreas. Okay, these acinar cells releases the enzyme component of the pancreas. Whereas the ductal cells, they release the bicarbonate. Okay, ductal cells release the bicarbonate. Acinar cells release the enzymes. Enzymes are fat digesting enzymes, protein digesting enzymes, and carbohydrate digesting enzymes. Fat digesting enzymes, protein digesting enzymes, and carbohydrate digesting enzymes. These are secreted by the ductal acinus cells of the exocrine pancreas. Okay, exocrine pancreas secretes fat digesting enzymes, protein digesting enzymes, and carbohydrate digesting enzymes. Whereas the ductal cells, they secrete the bicarbonate ions, water and bicarbonate. Okay, copious secretion of water and bicarbonate. So these are released into the lumen of the ducts. Okay, and through the pancreatic ducts they are released into the second part of the duodenum. So this is the second part of the duodenum. So it is released into the second part of the duodenum. Bile as well as pancreatic secretion, they are secreted in the second part of the duodenum. Okay, so when acidic chyme comes here, the pancreatic secretion comes. Okay, both the bicarbonate rich secretion and the enzyme rich secretion, they come into the second part of the duodenum, and it the enzyme component, they digest the proteins, whereas the bicarbonate component, okay, bicarbonates, they neutralizes the acidic chyme that arrives in the second part of the duodenum. Am I clear? Am I clear?
Yes, sir.
Yes, sir.
So there is the two part in the pancreas, right? One is the endocrine part, another is the exocrine part. Endocrine part are the pancreatic islets of Langerhans that release the insulin, glucagon, okay, insulin, glucagon and somatostatin. Whereas the exocrine part of the pancreas, it consists of two types of the cells, acinar cells and ductal cells. Okay, these are the acinar cells and these are the ductal cells. So acinar cells secretes the enzyme, okay, carbohydrate digesting enzymes, fat digesting enzymes and proteolytic enzyme. Whereas the ductal cells secrete the bicarbonate, okay, and water and bicarbonate, which is highly alkaline. So whenever the acidic chyme comes into the second part of the duodenum, that acidic chyme is neutralized by the bicarbonate. This bicarbonate is released in response to which hormone? Secretin. Okay, secretin stimulates the ductal cells to release more amount of the bicarbonate, right? And cholecystokinin, it acts on the acinar cells. It acts on the acinar cells to increase the enzymatic secretion. So endocrine part is this one, pancreatic islets of Langerhans. This one, okay? Pancreatic islets of Langerhans is the endocrine part, whereas these acinar cells and the ductal cells, they are the exocrine part. This one is the endocrine part. Okay, pancreatic islets of Langerhans, they are the endocrine part, and these acinar cells, they are the exocrine part. Okay, acinar cells and the ductal cells, they are the exocrine part. So endocrine part secretes the insulin, glucagon and somatostatin, right? Insulin, glucagon, somatostatin, whereas the exocrine part secretes the enzymes and bicarbonate rich secretion. Okay, enzymes and bicarbonate rich secretion. So function of the endocrine part is it helps in regulation of glucose balance, right? Endocrine part of the pancreas, it helps in regulation of the glucose balance. Okay, endocrine part, it releases the insulin and glucagon. So if glucose level is high, insulin is released, that will increase the uptake of carbohydrates, fats as well as protein, right? But mainly protein, mainly carbohydrates. Okay, if glucose level is high, insulin is released. Insulin mainly on the acts on the glucose to cause entry into the cells. Okay, it causes the uptake of glucose into the cells, whereas glucagon, it increases the glucose level. Okay, it have the uh, complementary action, right? Opposite action. Insulin decreases the blood glucose level, glucagon increases the blood glucose level. Somatostatin inhibits the insulin release as well as it inhibits the glucagon release, right? So that is the endocrine part. It will be discussed in your endocrine physiology, right? Endocrine part, it will be discussed in your endocrine physiology. But now importantly we have to discuss on the exocrine secretion of the pancreas. So exocrine secretion of the pancreas, it is secreted from the pancreatic acinar cells and pancreatic ductal cells. So pancreatic acinar cells secrete the enzymes, whereas the pancreatic ductal cells secrete the bicarbonate. Okay, these acinar cells, they secrete the carbohydrate digesting enzymes, fat digesting enzymes, and proteolytic enzymes. Pancreatic ductal cells, they release the bicarbonate, right? Bicarbonate and water. So pancreatic secretion, it consists of the high concentration of the bicarbonate, whose purpose is to neutralize the acidic sign that reaches the duodenum. So it contains the essential enzymes for the digestion of protein, carbohydrate, and fat. So these digesting enzymes, they are present in pancreatic acinar cells. These bicarbonate rich secretion, they are released from the pancreatic ductal cells. Okay. So secretion in the small intestine is pancreatic secretion, bile, small intestine secretion, and bile secretion. First, let's deal about the pancreatic secretion. So pancreatic secretion composition, we have to know about the composition and role of secretion, role of control of pancreatic secretion, and phases of pancreatic secretion. So composition of the pancreatic secretion. Okay, so pancreatic juice, it is highly characterized by high volume. Okay, it is high volume. It have virtually the same concentration of sodium and potassium as that of plasma. Okay, sodium and potassium concentration as that of the plasma. Then it has about 5 times greater the bicarbonate concentration than that of the plasma. Okay, 5 times greater the concentration that is in the plasma, right? Then it have much lower concentration of the chloride, and it is isotonic. Okay, the secretion that is released from the pancreas, it is isotonic to the plasma because there is the secretion of the water as well. Okay, not only the bicarbonate but also water is also secreted, so it is isotonic to the plasma, and it secretes the enzymes, pancreatic lipase, pancreatic amylase, and pancreatic proteases. Okay, so this is the main composition of the pancreas. And the pH of the pancreas, it is alkaline pH. Okay, it is the alkaline pH. It is important for your MCQs also, right? It is important for the MCQs also. The highly alkaline pH is present in salivary secretion, pancreatic secretion, gallbladder secretion, and gastric secretion. So you have to put a tick on the pancreatic secretion. Okay, it is highly alkaline. So composition of the aqueous component in the pancreatic secretion, it varies with the flow rate. Okay, when the flow rate is low, the pancreas, it secretes the isotonic fluid, which is rich in sodium and chloride. Okay, when when the flow rate is low, the pancreas secretes the isotonic fluid, which is rich in sodium and chloride. When the flow rate is high, okay, when the flow rate is high, that means when there is more acidic chyme is coming, then the pancreas secretes the secretion which is rich in bicarbonate. Okay, if there is less acidic chyme is coming, then it will secrete the composition which is rich in sodium and chloride. Whereas if there is the high flow rate, then it will secrete the composition which is mainly rich in sodium and bicarbonate. But despite the flow rate, the pancreatic secretion is always isotonic to the plasma. Okay, despite the flow rate, it is always isotonic. When flow rate is high, then also the secretion is isotonic. When flow rate is low, then also the secretion is isotonic. So ductal cells secretes the bicarbonate and water. This is the aqueous component. So it becomes isotonic to the plasma. Acinous cells, they release the enzymes. Okay, the enzymes are proteolytic enzyme, lipolytic enzyme, and carbohydrate digesting enzyme. Okay. So bicarbonate ions and water, they provide large quantity of alkaline ions to neutralize the acidic chyme. Okay, so uh, as the chyme that is released into the second part of the duodenum, it is highly acidic. Okay, it is highly acidic. So that acidic chyme, it is neutralized by the bicarbonate. This high acid, high proton concentration, it is neutralized by the bicarbonate. It converts the acid into the carbonic acid, then ultimately into water and carbon dioxide. Water, it is water and carbon dioxide, they are absorbed. Okay, they are absorbed by the small intestine. Right? So if there is no bicarbonate, then this hydrogen ion, it will erode the mucosal membrane of the duodenum. Right? So due to the presence of the bicarbonate, the hydrogen ion, they are captured because it is the positively charged, it is the negatively charged, so they are attracted towards each other, and it forms the bicarbonate, it forms the carbonic acid. Okay, hydrogen and bicarbonate combines together to form the carbonic acid. Carbonic acid is converted into water and carbon dioxide, which is both absorbed by the intestinal cells. Okay, and the pH is 8 to 8.5, which means it is highly alkaline. Okay, it is highly alkaline. So there are different enzymes present in the pancreatic acinar cells. Pancreatic acinar cells releases different enzymes. So these are the uh, proteolytic enzymes: trypsinogen, chymotrypsinogen, procarboxypeptidase A, procarboxypeptidase B, proelastases. They are proteolytic enzymes, but they are released in the inactive form. Okay, they are inactive. Once they come and they get contact with the cross border enzymes, they are converted into active form. Trypsinogen is converted into trypsin. Chymotrypsinogen is converted into chymotrypsin. Procarboxypeptidase is converted into carboxypeptidase, like this. Okay. Then there are the starch digesting enzymes also. Okay, starch digesting enzymes also. So that is the pancreatic alpha amylase. There are the lipid digesting enzymes also. Lipase, non-specific sterase, prophospholipase. Okay, these are the lipid digesting enzymes. There are the nucleases which digest the deoxyribonucleic acid and ribonucleic acid. And there are different regulatory factors: procolipase, trypsin inhibitors, and monitor peptides. Right? So the pancreas, it consists of the proteolytic enzymes. Okay, it consists of the proteolytic enzymes, but also pancreas is not autodigested because there are the presence of the trypsin inhibitors. Okay, this trypsin inhibitors inhibits, or it inhibits the conversion of of these precursor proteins into the active form. Okay, these enzymes, they are only activated when they reaches into the second part of the duodenum. Okay, these enzymes are activated whenever they are released into the second part of the duodenum. Otherwise, if they are activated within the pancreas, then it will cause the pancreatitis. Okay, it will cause the pancreatitis. Right? It will autodigest the pancreas. So formation of the pancreatic secretion. So like the salivary secretion, the exocrine pancreas also resembles the bones of the grapes. So they are made of mainly of the acinar cells and the ductal cells. So look at this figure. The acinar cells, they secrete the sodium, they secrete the potassium, they secrete the bicarbonate, and they secrete the chloride. Okay. The acinar cells, they all secrete, acinar cells secrete these ions into the lumen of the duct. And once it goes into the lumen of the ducts, the bicarbonate is secreted from the ductal cells, and the chloride is absorbed. Okay, chloride is absorbed. Bicarbonate is secreted, chloride is absorbed. Not only these are secreted, the acinar cells secrete the enzymes as well. And this is ultimately released into the second part of the duodenum. Okay. So this is the pancreatic cells. Okay, this is the pancreatic ductal cells. Ductal cells. So in the ductal cells, there is the in the basolateral side. In the basolateral side, there is the active transport occurring between the sodium hydrogen and sodium potassium pump. Okay, these two pumps are operating in the basolateral side, whereas in the apical side there is the action of the passive diffusion. Okay, there is the action of the passive diffusion, that is the chloride bicarbonate exchange, okay, antipode channel, that is the cystic fibrosis transmembrane receptor protein which is present, okay. This is the mechanism of secretion of bicarbonate from the pancreas, okay. In the pancreatic ductal cells, the water is combined with the carbon dioxide and it is converted into the carbon and bicarbonate. It goes to the lumen of the ductal cells in exchange of the chloride, okay, in exchange of the chloride, right? This is also the same figure, right? So carbon dioxide and water, they combine together, carbonic acid is formed, carbonic acid is dissociated into the bicarbonate and hydrogen it is exchanged by the sodium hydrogen exchanger, okay, sodium hydrogen exchanger, and in the apical side there is the bicarbonate chloride bicarbonate exchanger. So highly rich bicarbonate secretion is secreted from the ductal cells, okay, highly rich bicarbonate rich secretion is secreted into the ductal cell, and there is the paracellular transport, okay, paracellular transport of sodium and water also, okay, paracellular transport of the sodium and water also. And this chloride it is transferred from the cystic fibrosis transmembrane receptor protein into the lumen, and it is again exchanged with the chloride and bicarbonate. Now stimulation of pancreatic secretion. So pancreatic secretion, stimulation of the pancreatic secretion is mainly by three. One is the secretin, one is the cholecystokinin, and third one is the acetylcholine, okay, from the vagus nerve, right? So secretin, it is secreted by the S cells of the duodenum when there is the acidic chyme in the duodenal lumen, okay. When there is acidic chyme in the duodenal lumen, secretin is secreted from the S cells of the duodenum. So it acts on the pancreatic ductal cells and it increases the bicarbonate rich secretion, okay, it increases the bicarbonate rich secretion. So the second messenger involved here is cyclic AMP. You might have seen in this figure also, okay. Secretin, it acts on the receptor present on the ductal cells and it increases the cyclic AMP level. When cyclic AMP level is increased, protein kinase is increased and it causes the insertion of the CFTR proteins into the apical cells, due to which the bicarbonate and sodium chloride can be exchanged, okay. Then next is the cholecystokinin. Cholecystokinin, it is secreted by eye cells of the duodenum, okay. It is secreted by the eye cells of the duodenum in response to the small peptides, in response to the amino acids, and in response to the fatty acid in the duodenum, okay. Cholecystokinin, it acts on pancreas acinar cell, pancreatic acinar cell, as well as it acts on the gallbladder, okay. It acts on the pancreas as well as it acts on the gallbladder, because we have the secretion of fats in the second part of the duodenum, okay. Not only the peptides and amino acids are present, but also fatty acids are also present in the second part of the duodenum, right? And for proper digestion and absorption of fat, there must be secretion from the liver and gallbladder, okay. There must be secretion from the liver and gallbladder. So cholecystokinin not only activates the enzymatic secretion from the pancreas, but it also causes the contraction of gallbladder, so that gallbladder releases the bile into the second part of the duodenum, right? So cholecystokinin, it is secreted by eye cells of the duodenum in response to the small peptides, amino acids, fatty acids. It acts on the pancreatic acinar cells and it helps in secretion of the pancreatic amylase, pancreatic lipase, and pancreatic proteases, okay. So the second messenger system, it the second messenger system that is responsible for secretion of the enzymes is the by increasing the level of inositol triphosphate and calcium ion, okay. Increase intracellular calcium ion and inositol triphosphate increases the enzymatic components, right? Then third one is the acetylcholine. Acetylcholine, it is released in response to the hydrogen ion, small peptides, amino acid, fatty acids, okay. So it stimulates the enzyme secretion by the acinar cells, and it increases the effect of secretin. Okay, like cholecystokinin and secretin, it increases the secretion of the acinar cells. Okay? So there is the disease condition known as cystic fibrosis. Right? So, so it is the disorder of the pancreatic secretion. It results from the defect in the chloride channel that is caused by the mutation in the cystic fibrosis transmembrane conductance regulator gene. Okay, so so this condition is associated with deficiency of the pancreatic enzymes, resulting in malabsorption and steatorrhea. Okay, when there is no pancreatic secretion, then there will be no digestion and absorption of the proteins and fats. Okay, so which leads into malabsorption. It is not absorbed, and there is steatorrhea. Right? Steatorrhea means presence of fats in the fat in the presence of fat in the um, fecal matter. Okay, presence of fat in the fecal matter. Steatorrhea means presence of fats in the fecal matter. When fat is not digested, then the fecal materials, they become the like the mucus. Right? They become somewhat like the mucus. So this is the again the control of the pancreatic juice secretion. Okay, when there is the arrival of acidic chyme. Okay, acidic chyme arrives in the duodenum when there is the presence of the um, peptides, and when there is the presence of the fats, then there is the secretion of cholecystokinin, there is the secretion of secretin. Secretin is secreted by S cells, cholecystokinin is secreted by I cells. Secretin is secreted in response to acidic chyme, so it goes through the blood and it acts on the pancreatic ductal cells to release bicarbonate and water. The proteins and fats cause the secretion of cholecystokinin from the eye cells. They go through the blood. Okay, they go through the blood and they act on the pancreatic acinar cells. So the enzymes are released and they come to the second part of the duodenum. Okay, so acidic chyme is neutralized by bicarbonate. Okay, acidic chyme is neutralized by bicarbonate, and proteins, fats, and lipids, they are digested by the enzyme component. Okay, am I clear? Am I clear?
Sir. Yes, sir.
So look at this figure also. Whenever there is a decrease in pH in the duodenum, the S cell, secreting cell is stimulated. Stimulating cell stimulates the secreting release. Secretin increases the ductal bicarbonate secretion. Whenever the ductal bicarbonate secretion is released, there is the increase in alkalinity of the duodenum. Okay, chyme is neutralized. Okay, the acidic chyme is neutralized in the duodenum. Right? So when cholecystokinin is released, cholecystokinin, it causes the secretion of the enzyme component. So trypsinogen, it is converted into trypsin. Right? Normally trypsinogen, it is in the inactive form in the pancreas because there are trypsin inhibitors which is secreted by the pancreas. Okay, trypsin inhibitors are secreted by the pancreas, so it is in the inhibited form. Once it comes into the second part of the duodenum, there are the enteropeptidases that converts trypsinogen into trypsin. Once trypsin becomes activated, trypsin again augments the conversion of trypsinogen into trypsin. So it will convert the other enzymes, okay, chymotrypsinogen into chymotrypsin, and other enzymes, they are converted into the active form. Okay, this trypsin is converted into active form. So this trypsin, once it becomes more in the circulation, it inhibits the cholecystokinin secretion. Okay. So this is the I cells, that is the cholecystokinin secreting cells, right? So when there is the proteins, amino acids, fats, okay, proteins, amino acids, fats, they will activate the I cell secretion. Okay, cholecystokinin is secreted into the blood. Cholecystokinin goes through the blood. It will act on the pancreas and it will cause release of the enzymes. Okay, it will cause release of the enzymes. If the enzymes are released, enzymes, it will inhibit the cholecystokinin secretion. This is the negative sign. Okay, enzymes, it will inhibit the cholecystokinin secretion. Right? So look at this secretin. Right? Secretin, it increases the cyclic AMP level. Right? Secretin, it increases the cyclic AMP level. When cyclic AMP level is increased, protein kinase A is formed, which helps in fusion of these granules into the apical cells. Okay, right? Secretin, it causes increase in cyclic AMP level. Cyclic AMP, whenever it increases, it causes the translocation of the synaptic vesicles, uh, which consist of the pancreatic enzymes into the apical cell. Okay, secretin increases the cyclic AMP level and it causes fusion of the synaptic vesicles into the apical cell. Cholecystokinin, okay, cholecystokinin, it increases the calcium level. Okay, cholecystokinin, it increases the calcium level, and whenever calcium is increased, it causes fusion of the synaptic vesicles into the apical membrane, and the cells, acinar cells, they empty their contents into the ducts. So these are the, this is already I have told, right? So nervous regulation, regulation of pancreatic secretion. From the nervous regulation, acetylcholine and the enteric nervous system, they will increase mainly enzymatic secretion and bicarbonate secretion as well. Okay, but mainly enzymatic secretion, right? Nervous regulation, acetylcholine mainly increases the enzyme secretion, right? Mainly. Then secretin, it mainly increases the bicarbonate secretion. Right? Secretin, it mainly increases the bicarbonate secretion. Cholecystokinin, but it also causes the enzyme secretion. Right? It also causes the enzyme secretion, but secretin mainly increases the bicarbonate secretion. Cholecystokinin, it mainly increases the enzyme secretion, but there is little bicarbonate and water secretion as well. Gastrin also, it mainly increases the enzyme secretion and less amount of the gastrin secretion. So phases of the pancreatic secretion during the cephalic phase. Okay, during the cephalic phase, due to the smell, thought, and taste of the food, there is the about 20% of the pancreatic secretion. Okay, during the gastric phase, due to the vagal reflex and the enteropancreatic reflex, there is about the 10% of the pancreatic secretion occurs. Okay, pancreatic secretion has three phases: cephalic phase, gastric phase, and intestinal phase. Cephalic phase, gastric phase, and intestinal phase. So cephalic phase, in the cephalic phase, there is 20% of the secretion. In the gastric phase, there is the 10% of the secretion. Whereas in the intestinal phase, there is the 70% of the secretion. Okay, and intestinal phase, during the intestinal phase, the 70% of the secretion is due to the presence of the acidic kind, due to the presence of the peptides, amino acids, and due to the cholecystokinin. Okay, so this causes the about 70% of the pancreatic secretion. So intestinal secretion, it is approximately 1500 ml per day. Okay, 1500 ml per day is secreted by the mucosa of the intestine. Okay, so the goblet cells, they secrete the mucus-rich secretion. The intestine, they have the mainly two types of the gland: Brunner's gland and the Krebs of Lieberkühn. Okay, Brunner's gland and Krebs of Lieberkühn. So this is the secretion. Right? About 1500 boy, your 1500 ml is secreted from the intestine. So Brunner's gland, it is located in the duodenum. It secretes mucus. Okay. The function of the Brunner's gland is it helps in protection of the duodenal wall from the acidic chyme. Okay. It helps in protection of the duodenal wall from digestion from the acidic chyme. Then Krebs of Lieberkühn. Okay, they are the intestinal gland. Most of the small intestinal juice are secreted by the Krebs of the Lieberkühn. The pH of the small intestinal juice is about 7.4 to 7.8. Okay, 1 to 3 liters per day is secreted, and the secretion is stimulated by prostaglandins, vasoactive intestinal polypeptide, and acetylcholine. So small intestinal juice, it mainly contains the water, bicarbonate, digestive enzymes, enterokinases, amylases. The epithelial cells or the cross border cells consist of the peptidases, sucrase, maltase, isomaltase, lactase, intestinal lipase are present in the small intestinal juice. So function of the small intestinal juice is it supplies water medium. It supplies watery medium for the absorption of the substance. Okay, it supplies watery medium for the absorption of the substance. So this much for today. Bile secretion you will be doing in the hepatobiliary system, right? Bile secretion will be done in the hepatobiliary system, and this much for today. And mainly the function of the secretin and cholecystokinin, it is asked, right? Function of secretin and cholecystokinin has been asked. So you have to remember while asking, while the question is asked, you have to write down from which cells it is secreted, where it acts, what enzymes or what components does it release. You have to answer all. Okay. The Brunner's gland, they secrete the mucus. The mucus, it helps in the protection of the duodenal wall. Okay, because the acidic chyme is constantly entering into the duodenum. I know acidic chyme is constantly entering into the duodenum. So this Brunner's gland, it helps in the protection of the duodenal wall. Okay,
Yes, sir. Thank you, sir.
welcome.